ไม่ได้ยินเสียงจุมนา
ได้ยินไหมคะ
หมออุ๋ยไม่มีเสียงหมออุ๋ยต้องไปที่แชร์สกรีนแล้วบอกว่าแล้วก็เลือกแชร์ voice ด้วย
โอเคค่ะแบบนะคะโอเค so sorry ปิดอันนั้นไปแบบนะคะ
Could you see in the share screen to share the voice as well?
Okay. So, okay, I will show her screen. Okay, share the sound.
Yeah, okay.
Okay, you can see the screen, right? Sorry, start again.
Welcome to Marine Mammal and Sea Turtle Incubator. Distinguished speakers, participants, ladies and gentlemen, thank you very much for joining this incubator hosted by the Department of Marine and Coastal Resources, Thailand, with the support from the IOC WESTPAC. I would like to express my special thanks to our invited speakers for your participation despite your tight schedules. We really appreciate that. Marine mammal and sea turtle are the flagship marine species and the bioindicators or sentinel species that indicate the health of ocean ecosystem related to the human health. In recent years, there has been a growing concern about the decline in some marine mammal species and an increasing reported number for stranded animals. Countries in the region have been paying more and more attention to the conservation of marine mammals and sea turtles. This incubator will bring together researchers and other relevant stakeholders who are interested in the conservation of marine mammals and sea turtles, initiate a network for marine mammal and sea turtle population assessment toward the development of the UN Ocean Decade Action for the conservation of marine mammal and sea turtle in the region. I do hope you all have a comprehensive discussion and at the end of this 2-hour incubator, you could come up with the expected recommendations for research framework and direction in marine mammal and sea turtle that could help contribute to sustainable conservation of marine mammal and sea turtle and fulfill the knowledge gaps during the next decade. Thank you.
Okay. Okay. So, sorry. So I will thank you, Ms. Sumana, for the opening remarks. So we move to the first speaker. I would like to invite Dr. Tongkiat Kittiwattanawong to talk about the introduction of advanced marine mammal and sea turtle study to challenge. I would like to invite Dr. Tongkiat to talk about the introduction of advanced marine mammal and sea turtle studies to challenges in conservation management. He has been working as a marine biologist in Phuket Marine Biological Center since 1992. At present, he is a director of PMBC and has expertise in marine mammals and sea turtles conservation. Please welcome Dr. Tongkiat.
Good morning. Yes, it is quite a happy time to meet your old friends and new friends here. I would like to share my perspective in this UN Ocean Decade Kickoff Conference. As you know that this session is focusing on marine mammals and sea turtles. I would like to take this opportunity to thank online audience and colleagues for your attention and the future collaboration for conserving these animals. It is common that we think about protection when the loss has already occurred. My office also included at Phuket Marine Biological Center. Our interest in marine mammals started with the first stranded case 25 years ago. We have, from that time, we have expanded our stranding network all over the country. Until now, more than 6,000 marine mammals and sea turtle specimens have been collected and necropsied. By study of these specimens and their remains, we have gained more knowledge, little by little. And it's not only the knowledge, but more important things. We have fallen in love with this career, the job of marine endangered species research. And with this passion, we have looked into a very big question, how to protect these animals. The word protection and utilization are opposite actions. That is why we have the word conservation, in order to balance between the take and the keep. However, in the real world, it's not easy at all to balance these two things. The long-term sea turtle nesting statistic in Thailand might be an example. In these figures, reveal that the impact of, threats, dividing into 3 episodes. The first episode showed the sudden decline up to 85% within only 13 years due to the egg consumption. The second phase revealed that we lost additional 10% within 30 years due to the fisheries. And lastly, even in the past 30 years that we have implemented National Protection Acts, we still see not much improvement. In fact, the decline of sea turtle populations has not only been occurred in Thailand, but happening in other neighbor countries as well. The nesting statistic in Malaysia, in this case at Terengganu and Sarawak, revealed the decline trend over the same period of time as in Thailand, which is not so surprising because we know that sea turtles are highly migratory species. Sea turtle satellite tracking programs in both Thailand and Malaysia reveal the connectivity among populations within these regions. It can be concluded that no single country management plan is effective enough. Then the country should step in together, and together the management shall get success. Talking about conservation, let's begin with how to manage conservation. The first knowledge required is to understand about state or status. State can be indicated as population size or index such as population health and so on. The more interesting index is trend that show how the state has changed over time. The change can be decreased due to the threat or increased when we try to restore. The cumulative of both effects can be interpreted as risks, which is one of the conservation managing tools. Speciation is a process that living organisms have gone through their evolution to find their best fit for living. Thus, understanding of taxonomy is very important. The misidentification would give the wrong ontogenic understanding and lead to the mismanagement. The recent discovery of Ampan, the 3,300 years old baleen whales, raised awareness of how complex the baleen whales in Southeast Asia could be. The baleen whale in Thailand was thought to be a single species for decades. Until recently, the data derived from morphology and DNA reveal that there are more than 3 species. And with the new findings, we would expect more species to be discovered. As conservation managers, one should keep updating the information for better and proper management plan. The next question, and probably the most important question for conservation management, is how many animals are there? Where can they be found? And how does the abundance change over time? Perhaps the most common technique we use to estimate abundance of animals is distance sampling. However, the technique requires certain amount of survey efforts to gain probability detection function in order to calculate the abundance, not including several assumptions that can be easily neglected. In our department, we have responsibility to report national marine mammal status. Even though the distance sampling is a major technique, we also combine other estimation methods to fit the nature of animals and the limited resource and budget. In case of bright whales in the Gulf of Thailand, we have conducted photo identification. Mark and recapture then used to estimate population size. The data, along with voluntary sighting program, allow us to get on year-round population dynamic. Together with satellite tracking program and stranding network, make us possible to estimate the birth, mortality, and immigration rate of these whales. In case of dugong or child animals or even sea turtles, an area survey is the best and might be the only choice for population assessment. It has been more than 10 years that we have conducted our 10 years that we have conducted our survey for dugong assessment with small fixed-wing aircraft. However, it's quite expensive and risky. Moreover, it requires an experienced pilot to conduct good transit lines. Despite of fixed-wing aircraft, the ease of operating UAV or drone nowadays allow us to take video and photos. We can count the number, measure the size of animals, and map the distribution as never before. With photogrammetry, we can construct the size frequency graph that can be used as population health index. By combining data from aircraft and UAV, we have more in-depth and precise information, which is in return a better management plan. With the upcoming improvement of UAV endurance, this tool is very convincing survey technique. Back to basic, an interview was the first technique that Thai researchers employ for study marine mammals and sea turtles. We have gained not only information of the animals, but also receiving the perspective of local villagers. Thailand, together with neighbor countries, we were part of Dugong MOU team to develop standardized Dugong catch and bycatch questionnaires. With this method, we are able to get qualitative and quantitative Dugong information as well as spatial data. The technique allows researchers to get information over the large scale within a short period of time, and moreover, it was relatively low cost. Health monitoring program is part of population census for bright whale in the Gulf of Thailand. Body index along with other health index such as external parasites, wounds, and in this slide show the skin tattoo disease or STD, which is used as indicator of stress. Sometimes it was difficult to detect marine mammals directly. Passive cues may be used. This figure shows the feeding trail of dugongs over seagrass bed. The trail shows if dugongs are present or absent. The graph in the middle reveals that the length of dugongs can be estimated based on the width of trails. This allows us to understand the population structure. Seagrass recovery study on the right graph show that the seagrass took about 2 months for fully recover. Additionally, the carrying capacity of the habitat can be estimated based on the knowledge of seagrass and feeding trail. The passive cue can be obtained with more advanced sensing, such as acoustic logger that can monitor 24/7. The next generation DNA sequencing reveals an opportunity to detect these animals by only sampling the sediment and or seawater samples. The knowledge about connectivity among populations is another key issue for conservation. Understanding how the population relate to each other allow us to define conservation managing unit. In this case, the satellite tag is one of the candidate tools, but the information obtained from DNA analysis tend to be more conclusive. Based on mitochondrial DNA sequencing, as shown in this figure, the green turtle populations around the world can be divided into 11 managing units. However, this figure was only derived from the limited number of nesting stocks. I would like to encourage all colleagues, where possible, to collect DNA samples for a better understanding. Sometimes the population may be restricted to a certain area. The recent finding of dugong genetic diversity from the western coast of Thailand show the new haplotype that exists nowhere but this area. The discovery of this unique DNA raised the higher responsibility to conserve this dry gene dugong population. Necropsy is a standard protocol to investigate the cause of death. This job is similar to a detective that requires the look from both inside out about information obtained from the carcass and the outside in to get information from surrounding evidence. The figure shows the case of stranded dugong. Necropsy and 3D X-ray results show that the dugong was collided by an artisanal boat. Based on drift forecasting model and the condition of the carcass, we were able to track back the location where the incidents occurred. We encourage, where possible, to conduct necropsy for every stranded specimen. At the end of the day, the managing decision has to be made, perhaps with the best available information. This case, it is the only freshwater Irrawaddy dolphins population in Thailand. The bad news is this population is going to extinct. The recent assessment reported less than 20 animals in the lake. The major threat was gillnet entanglement, along with other problems such as breeding, inbreeding, populations, pollution, habitat, shallowness, and the decline of food source. The dolphins share the common source with many thousands villages around the lake. Even though the threat known, but it's not an easy choice to make decision. Lastly, I would like to say that conservation is an adaptive and long-term process. The knowledge that we have obtained might be only a tip of an iceberg. We then shall remember the mistake from the past and get moving together with a better conservation approach for a good fate of marine mammals and sea turtles. Thank you very much.
Oh, thank you so much. And I think we don't have a time for questions. Maybe we will answer the questions later. So thank you, Dr. Kongkiat, for your wonderful presentation. After we know about how important and interesting thing of these creatures, so next we move to our next speakers. Is a senior museum researcher who has over a decade of experience in marine mammal research and conservation in the Philippines. Please welcome Dr. Jo Marie Acibes.
Hi, good morning, everybody.
Good morning, John.
All right. Before I start, I should share my video and my screen. All right. Can everyone see my slides?
Yes.
All right. Okay. So again, good morning, everyone. Magandang umaga po sa inyong lahat. It is a pleasure to be invited to speak here today, and I thank the Department of Marine and Coastal Resources of Thailand for inviting me to participate. I am a senior museum researcher from the National Museum of the Philippines. I am here today to share some insights on conservation management of marine mammals and sea turtles based on my experience working in the Philippines and in the Southeast Asia region. Although most of what I will share I have drawn from the Philippines, I believe there are many similarities and parallels with other countries in this region.
Sorry.
It's all right. The Southeast Asian region is an area of high marine biodiversity, and this includes at least 36 species of marine mammals and 5 species of marine turtles. This is not surprising given the highly productive and rich marine environment with diverse habitats. Our diverse communities also have deep and rich cultures tied to the seas and oceans and the animals that inhabit it. It goes without saying that our nation's peoples have a close and complex relationship with the marine environment, which has evolved, changed over the centuries. As diverse as our nations are, so are the variety of management systems that we employ for the conservation of these animals. In recent decades, we have come to realize that our relationship with the sea, the ocean, and the organisms that live within it is an interdependence. And some may even say we need the ocean and everything in it more than they need us. What we do to the oceans, to marine animals, we do to ourselves. Hence, we need healthy seas, healthy oceans. So how are we doing in taking care of our seas, our oceans, and all these marine animals? Today, I'll be sharing some insights into the current threats to these animals in our region. I will also talk about some of the efforts researchers, scientists, and conservationists are doing to help protect and conserve marine mammals and sea turtles. I will also give some context on how some countries are managing these species based on the legal instruments and mandates of their respective local agencies. In recent years, particularly in the last decade, threats to these marine animals in our marine environment have become more evident that we can no longer ignore it. Pollution, habitat destruction, bycatch or accidental catch, ocean noise, climate change. These are probably the top threats. It is also important to note that many marine mammal and sea turtle populations are yet to recover from the impacts of long histories of capture or hunting. These are not just threats in the Southeast Asian region, but around the world. Who manages these animals? Who manages marine mammals and sea turtles, and how are they managed? Each country has its own management system and governing body that has mandate over the protection and conservation management of marine mammals and sea turtles. In Thailand, for example, as demonstrated by Dr. Kongkiat earlier, it is the Department of Marine and Coastal Resources. In Indonesia, marine mammals are under the mandate of the Ministry of Fisheries and Marine Affairs, but the Ministry of Forestry have also initiated programs for marine mammal management and conservation. In the Philippines, dugongs and sea turtles are under the Department of Environment and Natural Resources, or DENR, while cetaceans, whales, dolphins, and porpoises are under the Bureau of Fisheries and Aquatic Resources, which is under our Department of Agriculture. In addition to that, we also have the Biodiversity Management Bureau, also under our DNR, and the management of protected areas also falls under the mandate of the DNR. Taking the Philippines as an example, one challenge in the management of this species is the overlap of jurisdictions and often unclear mandates or responsibilities. All species of marine mammals and sea turtles are protected in the Philippines. And as mentioned earlier, 2 government agencies are mandated to ensure their protection and conservation. However, often the overlap of jurisdictions and gray areas and management makes enforcement of the law difficult. On the ground level, people get confused on who to report strandings or sightings or violation of laws such as capture of a dolphin or a sea turtle. And this may sometimes lead to delays in enforcement, or the information is lost among the maze of offices. Although the upside of this is the 2 departments often have to work together when dealing with this species. But perhaps what is more of a problem is that neither department has a regular long-term program that focuses on the research and conservation of marine mammals or sea turtles specifically. Most of the projects being conducted in the Philippines for the past, let's say, 20 or so years have been mainly conducted by NGOs or individuals affiliated with certain universities with funding coming from either foreign grants or foreign institutions. There is also a need for a national database for each animal group to serve as a repository of information and specimens, a database that can be used or accessed by government agencies, NGOs, research institutions, or individuals, a database that is open for access and can be utilized, evaluated, and even verified, not a database that is only collected by a certain group and only accessible to a certain group. In order to do proper conservation management of marine mammals and sea turtles, we need to overcome this research obstacle. And I think Dr. Kongkiat really described it quite perfectly about all the research that has been done, but still needs to be done, and all the challenges that goes with it, depending on the technology that you need to use. And of course, that goes without saying that funding is definitely part of that challenge. There's this paper that came out very recently, a few months ago, that looked into the patterns of research effort and extinction risk of marine mammals in the Philippines. Although the authors investigated the Philippines and focused on marine mammals specifically, I believe that their findings are not far from the situation in other countries in the Southeast Asian region and for other large marine species like sea turtles. One main point that they started out was global marine mammal research is disproportionately lacking compared to terrestrial mammal research and is strongly biased towards populations in Europe, North America, New Zealand, and Australia. And they go on in saying that research is still lacking. Again, as mentioned by Dr. Kongkiat earlier, particularly on topics such as population distribution, abundance, processes, and ecology. Research is also basically inadequate, and those that explicitly designed to address species conservation are few and far between. Continued monitoring of populations has not been undertaken since laws were passed protecting them. In the case of the Philippines, most of these laws were passed in the 80s or the late 90s. So that's a long time ago, yet there was never any monitoring of, again, as mentioned in the earlier presentation, any trends or changes in populations or distribution about what was known about a particular population of sea turtle or species of cetacean. It is important to document the consequences of what we believe are conservation actions in order to determine if efforts have been effective. This will also allow us to prioritize our actions, future projects, and the design of more cost-effective use of funding that is very limited to begin with. It can be said, compared to other developed nations, we in this region have been many steps behind when it comes to marine mammal conservation management. Yet, we have achieved so much in the past decade, especially. Some of the conservation efforts and achievements, of course, I'm only limited mainly on what I know about the Philippines. And some of them are, one, efforts have continued to expand network of MPAs, and some of them include parts of habitats of marine mammal species and some sea turtles as well. We have identified important marine mammal areas, marine key biodiversity areas around the country, all of which can be used as basis or justification for place-based conservation. For some species, research has progressed through the years, increasing our knowledge on the species and their populations, allowing for better decision-making and in mitigating certain threats. Through our Bureau of Fisheries and Aquatic Resources, we now have the Philippine Aquatic Red List Committee, and under which were formed subcommittees for various taxonomic groups, including cetaceans. Through this subcommittee, not only are local experts identified, but research and conservation projects funded by the government can be established and implemented. There are many more achievements and conservation success stories in the region, and I believe one factor of success is the support and collaboration among our nations. In 2013, the 3rd Southeast Asian Marine Mammal Symposium was held in Langkawi, Malaysia, and I believe a number of the people here in this incubator session were part of that. This symposium brought together researchers from around the Southeast Asian region to discuss various issues on conservation and biology of coastal, inshore, estuarine, and riverine marine mammals. It was also an opportunity to give updates on the status of species found in the region and how each country was doing in the research and management of these animals. As this was already the 3rd symposium, with the previous 2 held in the Philippines in 1995 and 2002, it was interesting to see how each country progressed from the 1st to the 3rd symposium. And I remember some of the senior scientists who were present in all 3 of the symposia commented that although there were significant progress in terms of knowledge on species and research technologies used, there were striking features that barely changed through the years. And this was mainly on the problem we faced and how we were dealing with them. I don't think I can say it any better, so I just quote Dr. Randy Reeves in what he wrote in the symposium report. He says, "We need to realize that not all conservation problems can be solved with more money, and that governance issues need to be resolved before a fix is feasible. For conservation to be effective, certain political and economic systems need to be changed, as does human demography issues that are also beyond the workings of scientists. There is a need for cross, transdisciplinary work when tackling conservation topics in order to manage such issues comprehensively." And he also says, conservation needs to be inclusive, reasonable, and practical. Finally, he ends by saying, in the end, it is important to avoid incessant negativity, It is much more effective to acknowledge improvements in conservation, celebrate the successes, and continue to encourage young people to carry the torch for this field. And I sincerely believe that. And with that, thank you very much. Salamat po. Kapnika.
Okay, thank you. Thank you so much. Okay, thank you so much, Dr. John. Wow, it's okay. We have another convener, Ms. Cha Kitchen Shampoo. Your turn.
Yes, thank you very much, Dr. John, for your valuable presentation. So now we know what you have been done from your experience and what we want going to do next. And what is the challenges and the gaps. So thank you again, and we move forward to the next presentation. Are you hear me? In the meeting room, can you hear me?
Yes.
Yes.
Okay. I would like to welcome Dr. Nantarika and Dr. Pacharapan, please.
Okay. Good morning, everyone. My name is Pacharapan Kamung. I am veterinarian from Phuket Marine Biological Center. And today I'm going to talk about marine animal species and standing management in Thailand. This is the outline of my presentation. First, we are going to cover the situation of marine species in Thailand. Then we will move on the standing network in Thailand, the conservation and management of marine species, and then the conclusion. So we are going to start with the endangered species in Thailand. Department of Marine and Coastal Resources is responsible for the research and protection of both the Andaman Sea and Gulf of Thailand. We have three main missions, which are population survey and estimation, sea turtle research and nursery, and endangered marine species research and rescue. For the MCR, we are responsible for a total of 28 marine mammal species and 5 sea turtle species. These are the examples of standing we have in the past. There are the variation of species from the small fillet point and the leatherback sea turtle to a live standing or sperm whale carp. With these standing events, a rescue center and standing network is needed for the success of marine life conservation. Therefore, due to the past standing and the abundance of marine resources along the coast of Thailand, Silitan Marine and Animal Rescue Center was found under Her Royal Highness Princess Siriwan Warina Ratasakanya Conserving Coral Reef and Marine Life Foundation. In this rescue center, we have rescued over 90 sea turtles per year. The main species we have rescued, rehabilitated, and released back to the sea are green turtle, hawksbill turtle, and olive sea turtle. For dolphin and whale rescue and rehabilitation, we have over 40 cases per year in the past. we have to improve with and remove. We have to improvise with movable pool or outdoor environments. But with the rescue center, we now have a facility for dolphin, whale, and dugong rescue and rehabilitation. This is the number of standing cases we had in a decade of our vision to protect marine resource and endangered marine species and expanding the standing network. In 2021, we have more cases in sea turtle with 60%. In dolphin and whale is 37%. In dugong is 3%. And proportion of standing animal between Gulf and Thailand in the slideshow. On 2021, from identified cross data, the cross of standing in Thailand, the sea turtle are mostly from human cross, mainly marine babies. In dolphin and whale are mostly natural case, mainly sickness and disease, as same as dugong. In the next topic about the standing network, I will give the floor to Professor Dr. Natalika Satsu from Veterinary Medical Aquatic Animal Research Center of Excellence, Chulalongkorn University. You're welcome.
Good morning, everyone. Thank you very much for inviting me to this symposium. For over 2 decades, DMCR have been collaborating with Chulalongkorn University to set up strong marine endangered species stranding network to help save these species. Our standing responses processes include various entities, especially the local people, NGO, and other concerning government offices. The system have been set up to handle both live and dead animals in various places. The first responders, mostly the locals, play the vital role in handling the stranding events. Therefore, we have put a very strong emphasis on keeping them informed so they can handle both live and dead stranding accordingly. After the first training course for DMCR staff in 2013, we have been expanded the training to other relevance group and 3 ASEAN countries. And the good news is in the last 4 years, we had 33 training courses for the local people, participants from 17 coastal provinces of the Gulf of Thailand and 6 provinces of Andaman Sea, together with NGOs and private sectors and government sectors. We can divide the participants into 5 categories. We are emphasizing very much on the coastal residents since they are our information sources. So if you look at the numbers, even though we have over like 18,000 participants, but the number is far from enough. We need more people. And when you look at the stranding network that we Thailand have been involved with, we are trying also not only the domestic, but the international organizations who have been working together and trying very hard to save these rare animals of the world. So we have been involved with organizations such as IWC, ISTS, the Asian Marine Mammal Stranding Network, Protector, and I have to thank the Philippines Marine Mammal Stranding Networks who have actually initiated for us in Thailand. So the master plans are divided into 4 areas that we are trying to work with. So the first one is that we are emphasizing on a species, trying to let them die less and then increase the population. And you all know that the most important thing is that we have to restore the habitat. And in order to do that, we have to look at the public awareness. We are trying very hard to convince and try to encourage the government and all the states' operation to try to involve conservation in their policies and their practice. And this is the dream that we are hoping to come true, that we can try to save the endangered marine animal status. We can manage the stranding. We can get all the national, international collaboration. We can try to protect the conservation marine area, and we can revive the population. And right now we are trying to prioritization on the species that has been important all over the world and in Thailand. And this would be the success in our conservation. For conclusion, I would like to add a note that in order to accelerate the conservation success, we are trying very hard to use advanced technology, as you have seen in Dr. Kongkit's presentation, to assist in the evaluation of marine endangered species status. We've been using aerial survey, drone, UAV, and acoustic tracking with satellite tagging and to monitor the population and dynamics. And the problem is, as you know, the cost. We are trying very hard to find that too. For the establishment of high standard marine endangered species rescued and rehabilitation center that we have set up in Phuket, as Dr. Pacharachupon mentioned, is now one of the top facilities in Asia. And we are building another one in Rayong with many local rescue centers, like small ones in the network. We are trying to have all these facilities to provide more support in our operations. Also, the advanced training is necessary. So we are trying to increase the training in marine forensic for veterinarians and the scientists. Also, we are trying to teach them how to make health assessment for animals in the field, which is very difficult and costly, as you know, but it is very important. We also conduct advanced research in sea turtle prosthesis, artificial reproduction, and sea turtle nesting conservation. And all of this, and the sea turtle prosthesis is the one thing that we are very proud of, because we have involved not only the biological aspect, but we also involve the aerodynamics, engineering, the material science, and everyone who would be helping us how to make the better prosthesis for the sea turtle that has been injured by boat, by fishing nets and everything else. So it has been very satisfying. And all these researches has resulted in several publications, especially in genetics research. Our mission, possible today, that we are really, really hoping that it will be a success, is trying to revive the declining population of Irrawaddy dolphin in Songkhla Lake and also in the Gulf of Thailand, and Bryde's whale and dolphins in the Eastern Gulf of Thailand, and finally the dugong in Trang province. To make this dream come true, we need our global collaborations, because we truly believe that together we can save them. And I would like lastly to present our success in our sea turtle prosthesis. Thank you very much. This is called the Extreme Plus Sea Turtle Prosthesis. This is the beginning of the research experiment. This sea turtle has been cut off, the arm has been cut off by the fishing net, and we finally got him to wear it with comfort, and now he can use it for feeding and also swimming. But of course, this sea turtle cannot be released because this is something plastic, but at least he can live happily with all the rest of them, and can fight for food or move happily inside our pool. I would like to thank the Department of Marine and Coastal Resources for making all this possible. Thank you.
Thank you so much, Dr. Nantrika and Dr. Pachapon. I think we can see a lot of work in the stranded marine and marine mammals and sea turtles, right? So the next topic is also very important and useful too, because it's about the animal health monitoring. He is a wildlife veterinarian with more than 30 years of experience in understanding the health of terrestrial and marine wildlife in temperate and tropical ecosystems. Please welcome Dr. Terry Work.
Hello, everybody. Thank you. Can you hear me?
Can you share your slide, please?
Yes.
It's very clear.
I'll share my screen. Hold on a minute.
Oh, cannot hear you.
Can you hear me now?
Oh, okay, I can hear you now.
You can hear me now. Okay, great. I'm going to share. Can you see my screen
Not yet.
Not yet.
now?
No, not yet.
Show again.
How about now?
Okay, it's
Great.
good.
Thank you very much for the Department of Marine Conservation to allow me to present this material. My name is Terry Work. I'm with the National Wildlife Health Center. We're based in Honolulu, Hawaii, and we work with biologists and managers to try to help recover endangered species in terrestrial and marine ecosystems. So some of what we do is technical assistance like training and outreach and necropsies of animals to determine cause of death. And we also do applied research, and really everything is done to help biologists and managers manage their wildlife resources. So, a lot of times we work throughout the Pacific. So some of these are the locations of US territories where we do work. And then also the blue locations, which are non-US places where we've also done some work on different wildlife species. We work on a variety of different organisms, marine mammals, sea turtles, seabirds, land birds, fish, marine invertebrates, and coral and algae. My focus today is going to be on sea turtles because that seems to be the focus of this workshop. And this gives you a map of the Hawaiian Islands. And the sea turtles in Hawaii, the major species we have are the green turtles. And they nest mainly in French frigate shoals. And then when they hatch, they go to the main Hawaiian Islands where they become adults and recruit to the nearshore foraging pastures. We don't really know where the pelagic stage is, where the immatures, when turtles hatch, they go out to sea for 5 to 7 years, and we really don't know where they go in the pelagic environment. So there's two ways to look at marine organisms. They have a nearshore phase, life stage, and they have a pelagic life stage. And stranded turtles are what informs the health of nearshore populations. And one of the big diseases one of the biggest causes of mortalities we have in sea turtles in Hawaii is marine turtle fibropapillomatosis. And this is this disease that causes tumors on turtles, external tumors and internal tumors. And in Hawaii, it's very unique. They also develop these oral tumors. So this is a photograph of tumors in the mouth, which causes all sorts of problems. And we spent a lot of time trying to develop tools to understand the pathogenesis and the ecology of this disease. So this disease is associated with a virus called cheloid herpes virus 5. And because we haven't until recently been able to grow this virus in laboratory, it's been very difficult to try to understand how this virus might play a role in causing disease. We can detect this virus by PCR, but we've had to develop other tools to really understand how this virus works in causing in disease pathogenesis. So one of the things we did is we had because we couldn't grow this virus in the lab, is we used molecular tools to sequence the virus. And then once we had the viral sequences, we could pick certain proteins of this virus to basically develop proteins and immunize mice with these proteins. And this allowed us to probe turtle tumors with this virus, with these antibodies, to show that this virus is actually associated with the tumor. So this photomicrograph on the left is a histology slide of a turtle tumor. And this photograph on the right is a slide where we've reacted this tumor with mouse antibodies to virus proteins. And you can see that the tumors are lighting up, showing that the virus is actually in the turtles. And by doing this technique, we're able to show that the way this virus gets around is it's shed in the tumors. So what happens is when the tumors grow, the virus grows in the tumors and sheds in the environment. And what's happening is that when you look at turtles with fibropapilloma, only about 20% of tumor turtles shed the virus. And they are accounting for 80% of the spread of this virus in the environment. So you have these super spreaders, these animals with tumors that shed the environment and shed the virus in large amounts in the environment and infect all sorts of other turtles. So these are super spreaders. We also know that with time, the immune system, as these tumors get worse and worse in the animal, these animals become immunosuppressed. And then as a result of this immunosuppression, they become colonized with opportunistic bacteria like Vibrio. So we found that Fibropapillomatosis leads to immunosuppression and opportunistic bacterial infections. Because we can't grow the virus, or we could not grow the virus in the lab until very recently, it was one of the really big things we wanted to know is what is the exposure status? When are animals becoming exposed and when are they getting antibodies to the virus? So in order to do that, we had to develop ways to detect turtle antibodies. So we raised monoclonal antibodies to turtle immunoglobulins, again, using mice. And then again, we looked at different viral proteins and we were able to express these viral proteins in what's called the baculovirus expression vector. So it's basically a way to grow the virus proteins in the lab. And using these reagents, we were able to show that we were able to detect the turtle immune response to the virus. So this graph here shows you increasing tumor severity in turtles. And on the y-axis is the amount of antibodies that these turtles produce against the virus. And as you can see, with turtles from Hawaii, where this disease has been very well studied, the more tumors they get, the more antibodies to the virus they develop. And in Florida, in the eastern United States, which is another area where this disease is present and has been very intensively studied, you can see that with increasing tumors, there's the virus, the antibody levels to viruses kind of flattens out and plateaus. And so that kind of shows that the immune response of these turtles in different geographic areas will differ according to the geographic area. So that really highlights that just because you see a disease like fibropapillomatosis in a given area, the animals in that geographic area may respond very differently to the virus, depending on the geography of where the disease is occurring. We recently were able to develop ways to grow this virus in the lab. So typically when you grow viruses in laboratory, what you do is you grow animal cells in the Petri dish, and then you grow the virus in the animal cells. But that technique has not worked with this virus in sea turtles. So what we had to do is we had to basically grow turtle skin in the lab in a Petri dish. So we seeded Petri dishes with fibroblasts. We then came back with turtle skin cells, and then we raised those cultures in the air-water interface. And by doing that, we were able to grow actual turtle skin in the lab. And by actually growing turtle skin in the lab, this allowed us to grow the virus. So this virus can only grow in a laboratory in a three-dimensional tissue culture matrix, which is kind of complicated to do, but it also shows that when you look at virus development in a three-dimensional tissue culture system, you can see the way the virus develops, which is very different than the way people normally look at viruses. And so this kind of highlights that if you really want to understand virus production and virus morphology and virus pathogenesis, you have to look at tissue culture in the three-dimensional system, not just in the flat petri dish with a sheet of cells. We also have an interest in what's happening in the pelagic environment, but with pelagic turtles, it's very difficult. Recall that we don't really know what happens with pelagic turtles when they hatch and go out to sea. So one way we can get a handle on that is we can look at turtles that are bicaught in the pelagic longline fisheries. And in the US, longline fisheries are required to bring dead turtles that they catch back to the mainland so we can do necropsies on them. And by doing that, we can see that pelagic turtles don't have any parasites and they don't have any fibropapillomatosis, which tells us that they probably acquire parasites and fibropapillomatosis once they recruit to the nearshore foraging pastures. We've also shown with another species of turtles that olive ridley turtles, that they have an infection with salmonellosis that causes renal disease in these animals. And this salmonella bacteria can cause significant strandings on the west coast of the United States. And we found that we also see these bacteriums and these lesions in the kidneys in pelagic turtles that are in the bycatch fisheries. And this is very unusual in the sense that this salmonella is very host specific to olive ridley turtles, which is kind of unusual for salmonella bacteria. So this has been a really quick overview of looking at tools that we develop to look at disease in sea turtles, but these tools can be applied to a variety of different animals. And this has been a collaborative effort among many institutions. And I'd like to thank you for your interest, and that's all I have. Thank you.
Okay, thank you, Dr. Terry Ward. Thank you very much for your interesting presentation. So I would like to move forward to next speaker. So because today we invite Dr. Ellen Hai from USA. Her research addresses the population and community ecology of threat and endangered species, incorporating local conservation efforts and regional scale coastal and marine management. And her presentation today are entitled The International Collaboration and Research on Marine Mammal by Catch. Please welcome Dr. Ellen Hai.
Greetings, everybody. Thank you, Pete Ong. And thank you, everybody. I'm so glad to see so many of you. And I miss you all. Stay well, everybody out there. Just have to say that. I've been lucky enough to work with the DMCR and many people here for quite a few years, I think since 1999. And I am honored to have been asked to speak at this Decade Action Incubator. I'll be talking about how it all began, how I started in my work, and all the wonderful people I've been working with, beginning with the work that I did when I first met Dr. Konkiat and Dr. Kanjana in Phuket in 1999, of course, and many others. But I'd also like to move forward from there through some of the work that we began working on the Western and then the Eastern Gulf Coast of Thailand, working with dugongs and Irrawaddy dolphins. and bringing together a lot of people to train from throughout Southeast Asia and South Asia. And I think at one point we had people from 10 different countries on our research trips. And to me, that is my dream, is to, like Dr. Conket said, and also Dr. Pacharapan and Dr. Nantarika, it is very important that people work together across national boundaries that, of course, these animals do not recognize. But then I'd like to talk about our project Getting to the Bottom of Bycatch. And to me, that really showed this kind of international cooperation. And there were some wonderful people, as you can see, from Vietnam, Thailand, Malaysia, Sri Lanka, and a few of us Americans, North Americans, who came along in creating a toolbox. And what we are is basically an interdisciplinary international team working to reduce marine mammal bycatch. And I want to, of course, acknowledge all of our in-country collaborators. Our study areas included the eastern Gulf Coast of Thailand and Trat Province, Kiên Giang Biosphere Reserve in Vietnam, the Cebu Tinggi Islands of Peninsular Malaysia, and Kuching Bay in Sarawak. And our rationale then was that there are these limited resources for scientists in many countries. We've heard that from Dr. John, and again from everybody. But we say that scarce data and poor understanding are no excuse for hindered progress. By the time we get all the data that we think we need, we might not have the animals left at all. So we need to work towards what I like to call a risk assessment, to identify, analyze, and evaluate the likelihood or probability of an event happening, and to look at its consequences. And what we did is we created a spatially explicit risk analysis in these areas where data were often inadequate. So the BIRA, the Bycatch Risk Assessment Tool, is a freely downloadable modeling framework that is based on the methods and applications from these two papers, which I'm glad to put in the chat for anybody, looking at our work in these 5 Southeast Asian fisheries. And BIRA can be used with a free QGIS or ArcGIS if it's available, and it's downloadable from the Invest Model Suite tools that are part of the National Capital Project out of Stanford University. And this first step is, of course, some sort of fisherman surveys or participatory mapping, or as we did in TROT, we had the formal line transect surveys or aerial surveys, or it could be from working together with experts or looking at the literature. But first of all, we need the sightings of the animals and the fishing locations and the kind of gear and the seasons used. And in Thailand and in Vietnam, we went around and talked to the experts who we were working with and other people from agencies and listed the different fishing gear that was used. The next step in BIRA then is creating a layer of species distribution. If we have enough data, we can use satellite data or animal data points gathered from interviews. In some cases, we use MaxSend as a suitability model. It's a presence-only model that's suitable for small sample sizes to model habitat use. And here is TROT. Here is Montrat. Sorry, I can't roll my R's. but here we're looking at habitat suitability in both on the left in the wet season and on the right in the dry season. And here also is our suitability. This is again for Irrawaddy dolphins in Kiang Giang, the Biosphere Reserve in Vietnam, where we had very little data and we had to depend on expert knowledge from people in various agencies and fisheries. The next step of BIRA is take the species layer and use it as input along with the stressors. Now the stressors then are defined as the gear-specific fishing areas, and we can define several or as many categories of fishing gear as we need to. And so here, again, this is on the left, the wet season in trout, and the dry season in trout. And these are from our 5 years of boat-based surveys. And again, in Vietnam, where there's very little data, we talk to many people in the Biosphere Reserve Office, fisheries offices, and people in commercial fisheries to generate these maps. So we do the best that we can, and fully to round out the model, we use a table for incorporating expert opinion and previous literature to rate the impact of gear to the species. So our gear ratings are based on both the exposure of the animal to the gear and the consequences of an animal getting caught. For the consequence, we have both sensitivity and resilience. For resilience, we assess the species and we look at understanding the biological factors that could make a population more resilient if bycatch were to happen to an individual. And that could be age, maturity, reproductive strategy, local species status, or population connectivity. So here's an example. Then let's look at surface gill nets. And this is generally what this table portion looks like for input into the BIRA toolkit. So we have our exposure, the spatial overlap of gear and animals, intensity, likelihood of interaction with species, and temporal soak time, temporal overlap, likelihood of capture by gear, and the current status of any management actions. Sensitivity would be the possibility of mortality to this animal if it does get caught, and the life stages affected by gear if known. And these require ratings from 1 to 3, and this is usually a conversation with local scientists. Now, based on the GIS layers, we have these 4 spatially explicit, these 3 spatially explicit criteria that we'll look at next. So the spatially explicit criteria that we use in most cases, and for the rest of this talk, I'll be depending on some work that we just completed off the northern coast of Peru, are maps based on fishing intensity, the likelihood, and like the fishing intensity is used as a proxy for the number of vessels in the water using gear at a specific time. The likelihood of gear species interactions is an overlay of the species distribution layer and the fishing intensity layer. And the temporal overlap and soak time is based on the number of hours fishing gear soaks in the water. So once we run this model using our gear ratings, we return to our stakeholders in each country and present these maps. Then we to see did we get it right? Does this look reasonable to you? Then we run the model to build our final synthesis, which is management recommendations. But before we really get into our results, we needed a way to address the uncertainty in our data inputs and outputs. We used the data standards based on Hines et al. 2020, where we worked together in a group, all of our Asian scientists, collaborators, to create a stoplight approach that delineated which criteria were needed to give us the best data, sufficient data, or caution and courage, because we really didn't have a lot of data. And what you can see is that in many cases, let's say for dugongs we looked at dugongs in the Sibu Islands off the eastern coast of Peninsula in Malaysia. We only had enough interviews for our animal sightings criteria and very little bycatch stranding data. And then our data output, our indication of uncertainty, was then put on each map. So again, for Peru, this will give you a good idea of the bycatch risk map. And this is for humpback whales in our study area. So as it appears, the entire study area does have bycatch risk, highlighting a higher risk nearer to the coast. This aligns with the whale's migratory route and higher fishing densities. Our stoplight shows green and yellow to signal low to medium uncertainty in the results. What I also want to point out here is that we're able to create subregions by distance from shore to show different results in, and also A, B, and C are different projected protected areas that people are proposing along the coast of Peru. So what we are able to show then are gear risk percentages for the subzones divided by distance from shore. So these graphs are split by gear type, with the zones going closest to shore, the artisanal fishery zone, similar to in Thailand, and the amount of risk associated with the gear. As you move away from the coast, there is less risk, but the highest risk is still from surface and bottom gillnets. Then Vira produces ratings for the exposure and consequence of possible gear interactions. While the humpback whales have similar exposure to most gear types, the worst consequences for the humpbacks are from bottom and surface gillnets, where they are mostly exposed to both gillnets and long leaves. For the three protected areas, there is some high risk in each zone, mainly contributing from gillnets. The medium risk is prevalent for bottom long leaves in Arrecifes de Puntesar in El Nido, but the Cursane is also pretty risky in the Banco de Mancora, which is further offshore. So these are results that we are able to get from the bycatch toolbox. And, oops, sorry.
Okay. And then
Yeah, can you hear that?
how many presentation you left?
I know, I don't know how to turn that off.
Yeah,
at this
I'm
point.
so
Oh,
sorry
wait,
that
yeah.
I have to disturb.
Okay,
Okay,
hold on
keep
one
going.
sec.
Yeah, please. Thank you.
Okay. Turn that off. There we go. All right, let's try that again. I apologize. So this
Yeah.
research, then,
Yes, it's okay. Okay.
okay, I'm almost done. I'll be in time.
Oh,
So
thank
we do identify these areas of high bycatch risk provides seasonal risk maps, initiates an organized conservation on bycatch, supports development of MPAs, uses low-cost methods, free software, and we provide local training, and values fisherman knowledge and input. To me, what's most important here are the conversations that we're able to have with local scientists, local fishermen, and people in different agencies. And here are just some of the workshops that we've been able to do in India, Malaysia, the Philippines, one of my favorite at Phuket, and in Vietnam.
Yeah.
Okay.
Thank you so much, Dr. Ellen. It's very good to have you here. And maybe the audience will feel like today we have all speakers that have very different experts. But however, we will come together today because we have the same goal. We try to conserve the marine mammal and sea turtle, but it came from the basic knowledge. So thank you so much, Ellen. So I would like to move. I would like to move to next presentation. This presentation from Dr. Dipani. She has been studying Humanoid dolphin in Chilica Lagoon in the eastern of India. So, and today is very special day for her because today is her birthday, and I would like to say thank you so much for pay your time for this meeting. Please welcome Dr. Dipani Sutaria, please.
Thank you. Thank you so much for inviting me and for wishing me, and I hope everybody has a wonderful day and the conference is successful. So I am going to start sharing my screen and hopefully I will know to do this properly. 1 second. Can you see my screen?
Yes.
Yes.
Okay, so I'll just enlarge it now. 1 second. Sorry. So all we seniors have learned how to use all this technology because of COVID. There. Okay, you can see my entire slide?
Yes.
Okay, all right. So thank you so much for inviting me to the conference. And as I was told that I should try and cover subjects that cover population estimation, I have just changed my slide. I've not gone into details of the methods, but I'm just giving you an overview of how things have happened in India using different lines of inquiry, different methods of inquiry. Okay. So we've recently found out, probably just in the past 2 decades, that there are at least 31 different species of cetaceans. I'm not going into dugongs at all in Indian waters. And as you'll see, we've had our first record of rhesus dolphins after researchers have been out of water. We've had our first records of Omuras whales, of the Arabian Sea humpback whales. And so I'll just go into the process of how we have been doing these things, and different projects are doing it in different places. Over here, you have resource dolphins, Irrawaddy dolphins, killer whales, striped dolphins, rhesus dolphins, and this is from east and west coast of India. So what are the challenges we face in India? I think South and Southeast Asia are very similar in terms of, our cultures, of course, our cultures in science, and then the kind of challenges we face. We are very, very, very similar. And so there's a huge gap in scientific data, as all the other talks have already said. And apart from, when I say scientific data, it's life history data, it's population stuff, it's, everything. We've not had cruises like the way the developed nations have had, like America and NOAA and all. When they do cruises, they have enough finances and enough logistics in place to carry them out. In India, we just don't have that. And sometimes justifying the cost of a large vessel survey is something that not all of us agree with. So we do have very huge gaps in scientific data collection. we also have these other things going on where we have gaps in legislation, we have mismatch going on between all the different agencies that are involved. In one side, wildlife and biodiversity protection, and on the other side, increasing the yield of fisheries, right? So there's a constant mismatch happening in the government itself. Then there is this lack of trust between people who are at the ground level or at sea all the time, basically fishermen, and administrating offices. There's a huge untapped potential of local knowledge systems. You know, this is a post-colonial thing, sorry, but it's just that we've lost so much of our traditional knowledge. And just about all our projects now include this because there's a lot of traditional local knowledge that even in terms of climate change, you know, people have seen out at sea how things have changed over the past 60 years. So people kind of know more if you'd ask them. And finally, in India, we still don't have any university or any institution offering any courses in marine mammal science. It's been in the pipeline for a very long time, but it's still not happening. Okay, next slide. So what are let's just look at only the first thing I talked about, which is the scientific data. And in scientific data, what are the kind of data gaps we use, which are really important for conservation? So the basic things are species distributions, diversity, the movement patterns, and space use over seasons, right? Then of course you want population estimation and trends and population size. After which is life history studies, prey preference and availability, growth rates, survival rates. Population structures using genetic tools, because is there bottlenecks going on? Is there a transmission between different isolated populations or not? So these are studies that are also required. And finally, stopping and mitigating incidental or target capture of cetaceans. So if you go back in our series of talks, I realized that, you know, Ellen was looking at the last point slightly. And there was somebody who looked at, from Hawaii, who was looking at life history studies and, you know, the stranding response network can lead to like this life history studies, prey preference, growth rate, and survival rates. So my next slide will then be only about the first and second data gaps, the population parameters, right? So what are we using? What do we use? We use two types of methods even for this. One is the qualitative method, which is your participatory informant networks or your interview surveys and your focal group discussions. So we use these even in India. And then there are quantitative methods, which are your line transect surveys, which are vessel or shore-based surveys using distance methods. There's mark recapture using photo identification surveys. And we also do passive acoustic monitoring, static and towed arrays. So we are trying to use all these at the moment in different locations. Different PhD students are using some of these methods. Just a glimpse into the kind of line transect surveys. We hire trawlers like this and we convert them into our platforms for doing distance-based surveys. And this method is now being used in, we also use ferries, inter-island ferries, which travel on the same route. So it's a uniform line. And so we use both those platforms of opportunity. And papers have been published using these methods in Orissa, in Andaman Nicobar, and in Lakshadweep, using distance-based line transects. This is something that was done in Cochin by Panikkar, where she was doing shore-based surveys at reticles and also had some very good data that she could gather on space use and dolphin density in a small area, which is an area you can see here. It is tourism, it is fisheries, it's shipping, it's CNG, it has all kinds of activities going on inside this harbor. And then there's a population of humpback dolphins that visit or spend time there. Then something that is close to my heart is the photo ID. We have not succeeded in using this method in any other species as yet in India, sadly. So it's been used only with the Irrawaddy dolphins in Chilka Lagoon. We tried doing this in Songkhla, but there were no animals there that time. And we have published, I've published from this, of course, the data. We had about 80 dolphins, and this is I think some of the best quality data one can have for closed or isolated populations that are well marked. Of course, all these methods come with their own list of assumptions that have to be met. So of course, if your animals are not well marked like these are, then using this method becomes very difficult. But this also is not just about population estimation. It can also be used for things like site fidelity, home ranges for each of these individuals, and then studying kinship and social structures also. So there's a lot of that goes into this method, and it does remain one of my most favorite methods to use. Then finally, this is something we've recently started probably in the last 4 years. This is Isha here studying finless porpoise. So her PhD is looking at how to customize your array and your methods for analysis to try and figure out the density of finless porpoises. And this is open ocean along the west coast of India. And she now uses a towed array. And this is some of the data that she has managed to find out. So as you can see, each color over here is a different individual. But visually, when we counted the animals, there were only 3 animals. And these are all of different bearings from underwater that the array got signals clicks from the finless porpoises. So this is a very good method. You have to, of course, have correction factors in place, and we have recently started this. But I certainly think that for elusive and rare species, passive acoustic monitoring is probably the best way to figure out what your actual density of animals is. This is looking at the blue whales and the humpback whales. This is also passive acoustic monitoring that we're using, not just for presence, absence, and space use. We now have recorders in 3 locations along the west coast of India. It's not just for presence, absence. If you're lucky, like if you look at the left side, these are blue whales. They have different singing notes. So what you see here is a singing note, and what you see here is a foraging one. And basically there are at least 2 individuals over here. This is what we found out from the passive acoustic monitoring. I will not put on the sound because it's a humpback whale singing. Then that's Abhishek over there, who started going out on fishing vessels to collect data on encounters with dolphins. What we do from these things is not only immerse ourselves in the fisherman's life and their way of living out at sea for 20 days, they spend time with us also. So when we say participatory informant network, when we say involving or including fishing communities, we don't mean just doing interviews with them. We actually mean going out and being with them and becoming part of their lives so that they can also become part of ours. And most of the people who do these things actually do it with, in India right now, are doing it with that perspective. So we teach, the fishermen know that there are these large pods of dolphins, but which one is which is what you can actually, even if you find one fisherman on the vessel of 20 who wants to start identifying, then it makes a huge difference to our data set. This is another way of doing participatory informant work where you're on the right. I'm sitting with fishing communities in Gujarat. Up over there, we are sitting with officers and dolphin tourist operators in Goa. So these are the kind of workshops and meetings we try to hold at least every 2 months with the communities. As you know, the country is huge, but luckily we have local teams working everywhere. So it's not like one team has to run around. And then we find out things like these. So there are whale temples in some parts of the west coast of India. We consider at least the large whales to be a blessing from God. And so collecting data for us has now for the large whales has now just started. And we are very thankful to the fishermen for actually taking the effort to do so. This is very cute. I would like you to hear it. You won't understand the language, but you might understand it's an interview going on. And at the end of it, I'm asking him, have you ever heard a whale when you were out fishing? And so this is what he sings for us. So what he was doing was he was trying to describe the sound of the whale, and then the fact that you don't have to be scared of them. They just come around the boat, they look at you, and then they go away on their own. So that is what he was saying over there. And then other participatory networks are also divers and free divers. So So if you can hear this, I'm not sure if you can, they're part of our network also. When he goes down, you'll hear a song. That was one of our first live songs recorded underwater. I hope you can hear. Yeah. And then this is a fishing community. This was just 3 days ago. We have a beautiful sighting from the fishermen of our first arrival of the humpback whale on the west coast of India. But you know, reaching this point in participatory work on such a huge area is a lot of work, a lot of effort. And the first and foremost is the well-being of the fishermen. Without their trust, these things would not be possible. So what do we do with all this kind of participatory data? As you would see over here, it was very difficult to figure out where to put the sound recorders, right? You might have money to buy sound recorders, but you've got to put them in the right places. So we used all the interview surveys and all the participatory informant network data and mapped these areas. And we decided that, well, we are not working in this area because of security reasons, but at least this area is where, and we have recorders in these 3 locations. So this is how we basically did not do the very financially heavy, logistics heavy boat-based surveys. I'm sure it needs to be done at some point, vessel-based surveys, but we did manage to put our recorders in the locations where we know the whales come across the Arabian Sea to India. So the utility of all these data sets that I have been talking about, of course, it's used in conservation and population status assessments in the IUCN, species protection status in India and the Wildlife Protection Act of India, regional level comparisons and collaborations for species like with the Arabian Sea humpback whale, at least for the migratory species, and then for the life history, if anybody is collecting life history data or at least looking at population genetic structures that can be compared across the species range. Very importantly, the biodiversity of assessments for marine systems. You know, when there's development projects, infrastructure projects going on, these kind of data sets are really important. So that, you know, even if they surpass all permissions and build a port, there is something in the background that people, if they ever go back and look for, is like there was data available, but we didn't use it. And finally, if most of you know about the important marine mammal areas atlas, it's the World Atlas. So we've provided data even for that, and we have quite a few IMAs in India. There are data gaps, as you can see, huge ones, and hoping to fill those up in the coming years. We also have now an online database, which is dynamic and which can be used. It is public, it's for public, can be downloaded and used by anybody if they wish to. The database gets data from fishermen and from researchers and from NGOs. So it's everybody contributing to the database. And so all these projects that I have described here, I thought I had only 10 minutes, so I was quite quick, are all these different people who are either already got their PhDs or are doing their PhDs or are working in NGOs. So I'm really thankful for all of them for taking all this work ahead in their own capacity and in their own interest and passion, actually. And my acknowledgements go out to a whole range of people, starting from the funding agencies who've been very kind all these years, to the dive companies, fishing communities, and of course, all the permission granting agencies. And that is it. Look forward to questions later. Thank you.
Thank you so much, Dr. Sutaria, for your sharing. It's very interesting topic, and we can get some point like a gap. So we move to the next speaker. He is the research professor and deputy director of the Key Laboratory of Marine Eco-Environment Science and Technology at First Institute of Oceanography. Please welcome Professor Shang Sulei. And I will say sorry because now time is very tight. So I need you to wrap up in 10 minutes. Okay, Sue. Thank you so much. Okay, please welcome.
Thank you, Matra. Can you hear me?
Yes.
Yes.
Yes, we can hear.
Okay, we see your presentation.
Okay, thank you.
Yes, yes.
Thank you. I would like to take this opportunity to introduce the proposal under the UN decade. The title is Smart Protection of the Marine Ecosystem. It's the idea from our teamwork. So I just the contact person and the leading scientist. So the background is on the UN decade of ocean science and to identify required knowledge, build capacity, and increase the use of knowledge, ocean knowledge and understanding, and also based on the global biodiversity framework post 2020 on SMART. And the background also comes from the technology revolutioning the study of organisms in their natural environment, not in our lab or in our catch. And the smart function of the marine ecosystem is to effectively attain knowledge on marine biodiversity, support conservation of marine ecosystem, and promote sustainable development of coastal society. We have proposed for two. One is both targeted on the icons of marine biodiversity. One mainly on the endangered marine mammals, and the other is also for the coral life health. So for the coral endangered marine mammals, we will integrate polyscience and technology, also with public wisdom. Firstly, for the endangered marine mammals, we will involve public wisdom by designing the apps, mobile apps for reporting the sighting events. This is many language varieties. Also, we use the new technology like marine environmental DNA to study the genetics by using the non-invasive sampling. And also we will use the UAV, as Dr. Gunkiat and colleagues mentioned already, as a novel height from the sky for the population size and behaviors and home range, so on. And also use the satellite telemetry to migrate and connectivity studies. Also for the distribution and habitat sensors, we will use more advanced technology such as the USV in the coastal area because of the limitation of the battery, and also on with the unmanned sailboat on the right bottom because it almost endless for and unlimited with power supply. It can be used both for the marine and coastal area sensors. For the coral life health, we will also use the species richness diagnosis from the environmental DNA method. And also we will involve public watching, public involvement by sharing the live images and the videos to the public from the on-site. And the merits of the smart protection of the marine ecosystem is to contribute to the decade 5 major outcomes, including a predicted ocean and an accessible ocean, an inspiring and engaging ocean, a productive ocean, and a healthy and resilient ocean. We have already made a consolidated foundation for our proposal. During the past years, we have been conducting the so-called MISTA project, the regional study of marine endangered species, including mammals and sea turtles in the tropical Asia for effective conservation and US and and also WESTPAC. We have developed what we call the VALA framework. The VALA framework borrows the idea conception from the traditional medicine, that is to propose the people's problem. We use the watch to the applic method like the sightseeing and photo ID, and ask like the social surveys to the local knowledge system, and listen using the acoustic, bioacoustic methods, and analysis of big data and samples and information to get the result. This is the framework we think we can wrap up all the existing and the developing methods, and can be addressed can be used to address the problem for getting data for the endangered mammals in the ocean. Also, our projects have various partners from this region, including not limited to colleagues from Thailand, like DMCR, Malaysia, like the UMT, IUM, IUMS, Indonesia, the Maritime University of Umrah, and Cambodia, Brunei, Bangladesh, Sri Lanka, partners from this outside of this region, and also China. We have been implementing our method on dugong, Irrawaddy dolphin, finless porpoise, bronze whale, and sea turtle studies. We have developed artificial intelligence for high throughput analysis of marine mammal images to assist our data process. Also, we developed artificial intelligence method for high throughput analysis of marine mammal sound. So both sounds and images can be processed and analyzed efficiently. And we have been using integrating the advanced platform by using the USV, the unmanned service boat, and UAV drones as a multi-platform to study dolphins and dugongs. Here are just two examples in Andaman Sea, in the Rang province coast, and also the Bay of Brunei. We have been using the UAV photograph to show the spatial displacement of dugongs and green turtles in their grazing ground. Also to present their population information on dugongs and green turtles. Also, we use the bioacoustic method from the clicking sound of the Irrawaddy dolphin to do their hotspots, both spatially and also temporally. Here I also present an example that's from the clicking sound from the Irrawaddy dolphin, we can infer their response to impacts of boat and information interference. We have also identified the common resource of Irrawaddy dolphin. And we use satellite telemetry to track various animals migration, such as the bronze whale in the Gulf of Thailand, and the turtles, the hawkbill from the Gulf of Thailand, and also the green turtle from the Andaman Sea area. And we have been using the DNA method to show the connectivity of green turtles from various management units. And our results show that the Bay of Brunei is a major foraging ground for green turtles from the Southeast Asia region, presenting its importance in the area. So that's our proposal, and we look forward to your support and welcome to you to join our cooperation. And I would like to welcome questions and comments. Thank you.
Thank you, Professor Zhang, for your nice talk and show a lot of new technique to conserve the animals. So last but not least, the network is the key for stranded animals rescue and providing the sightseeing of the cetaceans as well as sea turtle data. The next speaker is the president of the Philippines Marine Mammal Stranding Network and has been working on marine mammals since the 1990s. Please welcome Professor Lemuel Agornes. Okay, so can you see the slide, right?
Yes,
Okay.
we can see.
Let's start.
Good afternoon, ladies and gentlemen, or good day. Thank you for inviting me for this special talk. Today I'll be talking about the Philippine Marine Mammal Stranding Network, value of genuine engagement and networking. I am Lem Aragones, the president of the Philippine Marine Mammal Stranding Network and currently a professor at University of the Philippines, Diliman. First of all, the Philippines is an archipelago of 7,641 islands. So it's a relatively large island nation, archipelagic nation. And our nation is divided administratively, meaning for administrative purposes, into 17 regions, of which 15 have coast. And take note, the Philippines has a humongous amount of coastline. We have the fifth longest coastline in the world, amounting to about 36,289 kilometers. So you can see here the boundaries of the different regions in the Philippines. And in the Philippines, we are fortunate to have at least 30 marine mammal species, which 29 are cetaceans, whales, and dolphins. We do not have any porpoise, and the dugong. And these are the photos of some of the examples: spinner dolphin, melon-headed whale, rhesus dolphin, spotted dolphin, and the dugong. And they do strand. Okay, so unfortunately, they strand, and that's the reason why we formed the Philippine Marine Mammal Stranding Network. In 2003, there was an interagency workshop. It was held at Ocean Adventure, Subic. It is a marine theme park. In 2004, partnership between BIFAR, the Bureau of Fisheries and Aquatic Resources, and the owners of Ocean Adventure for capacity building and development programs for marine mammal conservation. And by 2005, its first project was to establish or to conduct the first national workshop on marine mammal stranding response. And in 2006, the second national workshop was also held in Ocean Adventure. Starting later part of 2006, we started doing the regional training, starting with Region 2. And by the year 2007, the expansion of the Philippine Marine Mammal Stranding Networks began. And by 2009, the establishment of the Memorandum of Agreements between PMMSN and regional offices of the Bureau of Fisheries and Aquatic Resources started. So what is the PMMSN? PMMSN is a SEC-registered group of professionals and volunteers throughout the Philippines committed to responding to stranded marine mammals, dead or alive. So our main objectives are to respond in the context of the individual stranders or mass stranded animals, in the context of their welfare or the animal welfare, and also to collect data. And that is These are the two main objectives of the network. The core organization members, meaning these are the ones that started, helped build, establish this network. The academe is the University of the Philippines Institute of Environmental Science and Meteorology, where I belong. The national government agencies such as the Bureau of Fisheries and Aquatic Resources, the Department of Environment and Natural Resources, an NGO, Wildlife in Need, and a private corporation, of course, as I mentioned, Oceanographic, who started to share the technology. PMMSN has been organized in various levels: regional, the provinces or provincial, and local government units, be it the cities or towns. Currently, we have certified 4,563 first responders. So these are the number of our certified members nationwide. So as you can see, we train and we continue to train, and we've been responding and we continue to respond. And that's the main context of the message of the genuine engagement that this network would like to share to the group. Here in this photo, you can see that I am demonstrating using a human example as a humanoid dolphin along the beach. And here is an example of a rough-toothed dolphin that we were rehabilitating. In this case, we rehabilitated this animal for 50 days before it was released. So the story is simple. We started training, and the training sort of was slow at the start. We were established in 2005, October 2005, first training. And so the request of trainings increased. These are the number of trainings per year. Okay. And right now we are already conducting the 123rd training. So you can see that out of these trainings through the years, we have acquired 4,563. And therefore, we've been training small groups ranging from sometimes 30 to approximately about 70. But in the advent of this pandemic, we've been able to increase it in the context of via Zoom as a webinar series. So these are the number of chapters. We have a humongous number of, but take note, 85, but 15 are regional chapters, and that's all of the coastal provinces have PMMSN chapters. And we've been very fortunate to deal with rare species such as the ginkgo-toothed whale here and Longman's beak whale. We've been able to produce manuals to enhance our training and our response, been able to develop a field guide to help out the responders. We even have been able to publish because the UP has been the research arm of this National Stranding Network. We've been able to conduct annually national symposia. And this is the main objective of this national symposia, is to share our experiences, build consensus, share results, and fortunate also, most importantly, to build the network. And right now, because of such a strong network, we've been able to almost record all the strandings nationwide. And you could see that started small numbers. This is probably an artifact of the people being able to recognize that they have to report, they've been educated to report the strandings. But somehow, starting 2014, more or less, the stranding incidents or frequency have settled. So we have that as an evidence of our stranding network growth. We've been able to also generate a powerful map using ArcGIS, using fish coral grids. to identify hotspots in the Philippines. These are grids of 15 kilometers by 15 kilometers. See the various color codes here for frequency of strandings in such area. We've been able to gather also a humongous amount of information, particularly the species composition of our marine mammals in the Philippines. And therefore, I would like to thank you guys, and also like to pitch in through this network, the Philippine Marine Mammal Stranding Network. We've been also able to assist people who have similar strandings in the whale shark as well as sea turtles. I hope you learned something today. Thank you. Enjoy the rest of the conference.
Oh, thank you very much, Dr. Lam. He may not here with us, but his presentation is very interesting and effectively. So we're lucky that we have 20 minutes left. So for this time, we have go to the discussion. We have the opportunities to listen to the experts from the marine mammal and sea turtle sectors. For discussion, our incubator go to define the important issues and recommendation about the future plan for conservation action. So I think we would like to listen from the floor, the question from the floor. If anyone has the question, please raise your hand. And because we also have the list of the topic that like a gap of our research in our regions. So the list of the topics is like research in the transboundary areas. So it's the one is so we are not to like a gap of data, and we don't have the project yet. Or the training program that can be improved our staff and our knowledge. So if no one raised the hand, I would like to ask the speakers, Dr. Kong Kiat, could you please give our recommendation for what kind of the research or what kind of the project we can do together for in the next 10 years?
Yes, I did not expect that questions for me. Actually, it should be convenient. Yeah, but as we all heard about that many speakers talking about research, talking about the things that they have done, and talking about what we should do next. I think by now it's just only that how can we have a platform or any mechanisms that we can share the data together, and those data can be used in common for all of us for conservation management. So that is the key point that we should have, I think. Thank you.
Thank you so much for your recommendation. I think if no have the audience to give the question, I would like to ask the next speakers, or Dr. John, could you please give us recommendation for our next
step?
Yes, thank you.
Hi, sorry.
Yeah.
Yes. I guess the only thing I could recommend at this stage is, like, if we continue what we have been doing in the past decade, we have been very good, at least in our small network in Southeast Asia. We've been very good at keeping connected, staying connected, and organizing workshops and trainings together to be able to enhance our knowledge and skills in marine mammal research and sea turtle conservation. For example, I guess we've been talking about this for some years now. Having the next CMAM symposium could be one.
Yes.
One recommendation I think that I could give.
Yeah.
I hope it happens soon. Yes, the pandemic is still upon us, but maybe in the next year or so, it could finally happen.
Yeah. Well, very good, because I think if we propose this the next step to UN, maybe they can one of our support or some sponsor our conference, CMAM conference. I will highlight this one also. So next one, please, Dr.
Nantrika.
Dr. Nantrika, please give us the recommendation.
Thank you very much. I have to close. Can you hear me now?
Yes. I would love to see your face without mask.
Okay. First of all, I would like to congratulate on the success of this symposium. But one of the problems that I have noticed is that a lot of people would like to do research, but we have problems on the connectivity between the boundaries. That is that we have to try to set up some meeting with scientists and try to make sure that, to send samples across the countries for research should have some kind of special exception, because this has been really, really difficult. And the country like Thailand, we have a lot of equipment, but we probably will need more help from other countries. And also countries that just began this stranding network like Vietnam or Cambodia or Myanmar. They can also benefit from this because they can send samples to us and we all can do this research together. And I think for the topic of research, I'm sure certain countries have the definite problems that they are interested. And so what Dr. Kongkiat has mentioned, the database is very, very important. Right now, we have good connections. We are all friends and we have this WhatsApp or, all these discussion groups going on, but it is not tangible. So I would like to suggest that if you can have some kind of center, which is like an IT center, where everyone can put in the data from their countries or their problems that they would like to be solved by the neighboring countries, something like this, and this would be a real strong network. One of the things that I have noticed is that a lot of times when stranding happens, there's not enough people in that countries or enough experts. We need the central funding to try to send people across, just because we all don't work for money anyway, we have passion. So what would be nice if you can get like private sectors or some kind of organization to fund for the traveling and food. That's it. That's all we need. And our team or your team can come and help us, and we can go and help you, as soon as possible. Because all this time that we discussed over the social network, mostly the carcass has been decomposed and we get nothing. So time is very important. Thank you.
Thank you very much, Dr. Nantarika. So I pass to Dr. Pacharaporn. Do you have any recommend? Or maybe if not, Dr. Ellen, please. Are you still here with us? Thank you.
Hi, everybody. I totally agree with what's been said before. I think the CMAM is important and is a lot more action than just the WhatsApp I think we should keep training each other. I'd like to see if you're going out on a survey, invite people from different countries. I'd like to see maybe other people. I mean, Dipani is doing such amazing stuff on a shoestring and really working hard to create a new cadre of Indian researchers. I'd like to see those researchers invited to other countries, or maybe Dipani's group inviting someone from Thailand or Malaysia or Vietnam or Cambodia. I think we need to really share our knowledge in very direct ways out in the field. And Dr. Concat is totally right that database sharing is, I believe, critical, because, you know,
Yes.
especially looking at the oceanography in this area, these are not the animals' borders. Thank you very much for this opportunity.
Thank you so much, Ellen. So, Dipani, please give us the recommendation.
You unmute your microphone first.
Yeah, I don't know. I started off after I finished my master's on dolphins. I met Brian and Ellen and all of them, and I knew there was a whole world out there of researchers. So one of my beginnings of all kinds of field research happened in Thailand at the Phuket Marine Biological Science Institute. But I find it very difficult to actually recommend something for another region. But I do agree about building capacity. And when I mean building capacity, I don't mean that I go to Thailand and do something and then the people there are only writing up and they're not really doing the research or not doing the analysis, you know. When we say build capacity, we say build the capacity of that person so that they can carry on this work themselves. So that is the way I see capacity. I think having the next CPAN conference, sharing data or at least sharing results so that you can publish together, because data sharing is easy to say but not easy to do. Not just because of personal or professional reasons, but also because of national regulations, right? So we should at least be able to use similar methods so that our results are comparable and useful for conservation. But yes, build your capacity so that, you know, in the long term, you don't have to use fossil fuels for somebody else to come do the work. That's my fossil fuel is a big thing. So yeah, that would be my thing.
Yes. Next one, please, Dr. Thierry. Are you still with us?
Yes, I'm still here.
Thank you.
And yeah, I would endorse what everybody else has recommended. I do agree that foreigners coming to other countries and doing a little bit of work is not as productive, I think, as getting people in those countries up and running with these techniques. I'm happy to say that we have a student from Thailand who's working with us and doing a PhD. So we are very pleased with that opportunity, and we hope that once she finishes, she will come back to Thailand and do great things for wildlife health and sea turtles and coral reefs. So thank you very much for this opportunity.
I have a question for Dr. Terry. As the veterinarian, so I think the regional disease rank in this region is not enough. Do you have some recommend for this issue?
For wildlife health?
For the disease surveillance in the sea turtles or in the marine mammals, I
can say that I can say that the wildlife health investigation capacity has been very useful in Hawaii and the Pacific for recovering endangered species. We're very good as biologists in monitoring things. We're not very good at getting answers and intervening to recover populations. And I think that that's where wildlife health could really play a very important role. And there's some very good examples for birds, coral reefs in Hawaii and the Pacific, where wildlife health investigations have led to significant management, positive management interventions. So I would encourage everybody to think about more broadly than just sea turtles and marine mammals. It's applicable to both terrestrial and marine ecosystems.
Thank you so much.
Yes, the last speaker, please, Professor Sule.
Thank you. Can you hear me?
Yes, yes,
Ah.
you can hear.
Yes, thank you. I would like to suggest that that nowadays we need to get a lot of data through many ways to come up with method or measures. So I would like to, the reason we propose the smart protection of the ecosystem is to free us from the labor and time intensive data collection into the mind map that we can spend more time and effort to come up with ideas and to conduct efforts to solve the problem rather than just rely on the labor intensive and time consuming efforts to wait for the stranding response. And I would like to suggest we do not only rely on the once there is a report, then we do some we conduct some response. We can, through the smart possession of the ecosystem, we can predict when, where, and what we should prepare to make measures of protection. Rather than we get alarm, then we go out, we get another alarm, then we go out again. So we keep using, setting off fires, and the effect, or while the effect is not ideal to us. The other thing is that I would like to suggest that we need to pay more attention to transfer our knowledge and results to the public, to let them know and accept the value and importance of these animals and ecosystem, and also to draw their support to our measures and to draw their active involvement in the conservation of the biodiversity and the ecosystem. So that's my suggestion. Thank you.
Thank you so much, Professor Sule. So we have time limit, and this time is the final, the last chance for us to summarize. So if the audience have a Q&A, you can leave your Q&A in the chat box, and we will reply your question via email. So for after finish the meeting, we will summarize our discussion and recommendation and send you all of you via emails. So I believe that this event is of a great importance to all stakeholders involved in marine mammal and sea turtle conservation. Initiate a network for marine mammal and sea turtle population assessment toward the development of UN decade action for the conservation. Before we close the conference, on behalf of Department of Marine and Coastal Resources, we would like to take this opportunity to sincerely thank all of speakers, audience, and our sponsor, IUN's WESTPAC, and Ministry of Natural Resources and Environment, Thailand, for make us together So I think we can close the conference like just says thank you, and I would like to take a photo, a cap for all of your speaker and audience. So please turn on your camera and just take a minute. Take a just okay. Okay, we have right now 88 audience. So we will take a photo for the first page. We have four pages. So for the first one, okay. 1, 2, 3, smile. Okay, next one is the page number 2. So next. Okay, the page number 2. Okay, 1, 2, 3. Oh, I'm waiting for the number 2 page. Okay, one more time. 1, 2, 3. Thank you. Next one is number 3. Okay. No one open. Okay. No one turn the camera on. So that's just the cap your name. Okay, that's fine. Okay, the last page. Okay, 1, 2, 3. Okay, thank you so much for joining us. Goodbye. Have a nice day.
Thank you so much.
Thank you so much.
Opening ceremony.
Happy birthday,
Dr.
Dr.
Deepak.
Yapani.
Happy birthday.
Happy birthday.
Oh, I'm so exciting that the first time for me. So first time for me and Dr. Wachara to conduct the convener. I would like to apologize for accident or something mistake.
Yeah.
Congratulations. It's been very
Congratulations.
successful.
Bye-bye.
Bye.
You did a wonderful job.
Our team, DMCR, Misu team, don't forget to report your, don't forget to give me the how to your record. Thank you so much. Goodbye.
Bye.
Still have some text